My name is Wijatyk, and today I will be presenting on behalf of Miltenyi Biomedicine. I am planning to share key learnings based on Miltenyi's experience from the scale-up required to conduct a pivotal clinical trial. So a few words about Miltenyi Biomedicine, who we are and what do we do. We are a biopharmaceutical daughter company of Miltenyi Biotech, developing personalized cell and gene therapies for patients worldwide. With the ultimate goal to improve and extend patients' lives. Miltenyi Biomedicine was established in 2019 and built on decades of research and development experience from Miltenyi Biotech in the cell and gene therapy area. I think you all know about Miltenyi Biotech. It's a well-known entity in the cell therapy space. So Miltenyi Biotech and Miltenyi Biomedicine are complementary and interconnected organizations. While Miltenyi Biotech focuses on discovery, research, and pharmaceutical development, GMP products delivery, and technology with prodigy process engineering, Miltenyi Biomedicine takes care of clinical development with the ultimate goal of registration of our products and then their commercialization. Let me now talk about our first product in clinical development, which is Xamtocell autoleucel or Xamtocell. So it is a bispecific CD20, CD19 CAR T cell therapy that we developed. So the manufacturing of the product starts with the fresh leukapheresis product that is delivered to the cell factory from the clinical sites. The leukapheresis product, the cells are enriched for CD4 and CD8. And then they are transduced with the lentiviral vector encoding a second generation CAR, CD20 and CD19, with the 4-1BB co-stimulatory domain and CD3 zeta domain. Then it's expanded with IL-7 and IL-15 and the Tex-Mex media. So the whole process of manufacturing takes 12 days. So on the slide at the bottom, you see the full construct of our CAR T transduced T cell. And the manufacturing of the process you can see at the right side of the slide. So what is important to emphasize that this process allows us to deliver the product to the patients within 14 days. So 12 days of manufacturing plus 1 day for the transportation of the leukapheresis product and 1 day for shipment back of the CAR T therapy to the patient. So this is a relatively rapid process that is a fresh in, fresh out, meaning fresh leukapheresis product comes into the cell factory and the fresh drug product leaves the cell factory to be delivered to the patient. So why we decided to develop Xamtocell? You may say the space is already crowded, there are multiple CAR Ts for the same indication. Our first indication is relapsed refractory diffuse large B-cell lymphoma, so similar to what's registered in that space. However, although the development of the CD19 targeting CAR T therapy represented the major accomplishment, and benefited number of patients, it does have some limitations. Notably, relapses do occur, and they occur quite often. It may happen even up to 50% of treated patients. Different mechanisms have been postulated to understand the relapse. Target antigen escape via CD19 loss or CD19 downregulation appears to be maybe the most common. So the dual targeting of CD19 and CD20 can potentially be beneficial and may help with the more complete B-cell ablation and reduce risk of tumor cell escape, reduce relapse, and improve patients' outcomes. Since we've seen very encouraging results from our preclinical data, we have started and we have conducted the phase 1 clinical trial, DALI-1. DALI-1 is a single-arm, multiple-center dose escalation study that was conducted mostly in Germany. So the data from this study shows preliminary efficacy and favorable safety profile. So as you see from the slide on the right side, we have achieved 75% of the objective response rate. If you take into consideration 2 dose levels, we have studied 2 dose levels, the 1.0 and 2.5 millions per kilogram body weight. So as you see, we had a good response rate, overall response rate. However, more importantly, in our group studied, we've seen 5 complete responders, and those 5 complete responses were maintained not only at 12 months, but also they continued up to 2 years that the study has reported the data on. We actually presented the poster at IHA this year in Vienna. So in terms of the toxicity, so we consider the product having favorable safety profile because we have not seen grade 3 or more CRS or neurotoxicity. Other adverse events seen were consistent with the profile seen with other CAR-Ts. So encouraged by this early data in patients, we have established the recommended phase 2 dose, which is 2.5 millions per kilogram body weight, and we have initiated 2 pivotal studies with Xamtocell. One study is being conducted in Europe. This is a study in the second line at DLBCL, and the other study is conducted in US, and that is for third line DLBCL. I am pleased to share with you the design of that study. So the DALI-2 US is a phase 2 single-arm study in relapsed refractory DLBCL in patients who failed at least 2 therapies. So the patient undergoes a screening process in order to qualify for the study. So they are, the inclusion and exclusion criteria are being verified. And after this is successfully completed, the patient is enrolled and undergoes the leukapheresis. So as mentioned before, the leukapheresis product is directly shipped to the cell factory and serves as a basis for manufacturing of the Xamtocell. So during that process, a number of steps, of course, are taken at the manufacturing site, and I will talk about that in a moment. But also, at the same time, the patient has to undergo the lymphodepletion. And the lymphodepletion, we allow for 2 regimens in the study. One regimen that is maybe more traditional one with the cyclophosphamide, total dose of 900 milligrams per square meter, plus fludarabine, total dose of 90 milligrams per square meter, given on day -4, -5, -4, and -3. Or the alternative regimen that can be used is bendamustine with the total dose of 180 milligrams per square meter, and that's given only 2 days. So once the cells are ready for the administration, they are being shipped to the clinical sites. They are shipped fresh, so fresh in, fresh out. And then the patient is administered the therapy as an infusion, 15-20 minutes lasting infusion, and then enters the follow-up of 2 years. So the primary endpoint of that study is the objective response rate at day 28, as assessed by central review using the Lugano 2014 criteria. And then we have number of key secondary endpoints, which may be the most important one at this point is really duration of response, because coupled with ORR, duration of response shows you not only that if the therapy is working, the patient is responding, but also it shows you whether the therapy is durable. That's super important for the single arm study. Of course, the overall survival is also important, but because it's a single arm study, this is not necessarily a way we can compare it to a comparator arm. So when we think about what does it take to initiate a pivotal trial in US, what do you need to have to start such a study? So there are a number of prerequisites, and the requirements are not trivial, as many of you know. So it starts with the preparation for the pre-IND. It's super important to get alignment with FDA. You need to have alignment on the CMC aspects, and you need to have alignment on clinical. So for CMC, the adequacy of the release criteria, tests, and the release plan are very important. This has to be aligned. The scope of analytical package is important, and comparability is super important. You see, we have a study, first study was done in Germany, so we have another manufacturing site in US, so we want to show that they are comparable. So if you have multiple manufacturing sites, you need to show comparability. So comparability, comparability, comparability. That's a key factor that needs to be agreed on with FDA. How do you do this? Then, very important thing, processes needs to be put in place to maintain the chain of the identity from the moment of leukapheresis collection through the CAR T-cell manufacturing and shipping to clinical sites. Of course, stability needs to be taken care of. You may also need to do a transport study to show that there are no major changes occurring when you are transporting your product. Last but not least, you need a good clinical package. So the pivotal study design and proposed patient population and the sample size, this all needs to be agreed with the FDA. You need to design the study in such a way that it supports your indication. To start a study or pivotal study, you need to show you have a sufficient safety data. So if maybe first in human study was on a smaller side, maybe there were some questions about it, then you need to consider to have initial lead-in phase maybe with a few patients that are being staggered for enrollment in this pivotal study. Last but not least, you also need to think about how toxicities will be monitored and addressed. So there has to be a plan for detecting, monitoring, and addressing toxicities in your clinical protocol, and stopping criteria need to be put in place in case unplanned or planned but in excessive number toxicities occur. So as I said, our product is fresh in, fresh out. So this requires a super strict organization. There is no wiggle room for delays, nothing can wait, patient is waiting. The lymphodepletion is happening on day -1, and from that time, the clock is ticking. We cannot not deliver the treatment to the patient. So, in a nutshell, this slide recapitulates what's been summarized, but I think the key message here is that needs to be a rigorously orchestrated process so the infusion can happen on time and within this 14 days needle-to-needle administration. Now I will spend a few moments to talk about our manufacturing process. So on the left side of the slide, you have all the steps listed that are required to deliver the drug product to the patient. So on day 0, we start with a leukophoresis product, which as our starting material needs to be reviewed, needs to be assessed for the adequacy, and then we proceed with the cell selection. So this is a standardized process with the CD4 and CD8 enrichment, and then we move to the activation process. For activation, we use our proprietary media, Max GMP T-cell Transact, and that leads to next steps, which happen between day 1 and then day 12, with the cell transduction using the lentiviral vector transduced with the CD20, CD19 CAR, progressing to cell cultivation using the Tex-Max medium, IL-7 and IL-15, and then final formulation happens using Clinimax formulation solution. And then the process concludes with the final fill of drug product. The dose that we deliver to the patients is 2.5 millions per body weight or per milliliter as well. And this whole process happens using the automated Clinimax Prodigy platform with the closed single-use tubing set. What is important in this whole process, of course, is to perform number of testing that have been agreed on with the FDA, and they are part of the material adequacy assessment. So when you look at number of tests that you need to do for safety, this includes microbiological examination, mycoplasma testing, endotoxin, and RCL. Why I crossed out the RCL from this slide is because in our process we were able to demonstrate after a certain number of patients that we have no issues with RCL. So now we don't need to test for RCL. And as you see from the right side of the results in more than 55 clinical runs, all the tests were passed. So it means we have a robust product that delivers the drug product to the patient in a reproducible and robust manner. Now, when we think about the CQA testing, again, part of the release criteria or in process testing, there are a number of steps that need to be assessed. So cellular composition, transduction frequency, vector copy numbers, and last but not least, we also look for potency using the interferon gamma assessment. From the right side, you can appreciate that our process is quite robust and reproducible. I think one thing that also is worth to emphasize that Miltenyi is actually uniquely positioned because we not only have this manufacturing platform, but we also have all analytical methods and media using for this development of our Xamtocell. So we are in a unique position to really scale up for the pivotal trial manufacturing because we have all these elements in place. Last but not least, QC and QA. Why this is super important, specifically for the Xamtocell? Because it's a fresh product, so we have no time to lose. So if we have no time to lose, we really need to have a super efficient process that allows us to conduct different assessments on a rolling basis and on an overlapping basis. So wherever we can, we need to gain efficiencies. We need to I would say, really fight with time to deliver the product on time to the patients. Moreover, there is a day 5, day -5 time clock for the patient. So we need, at that point, we need to assure the clinical site that everything is in good progress, that our product is looking good, and that they can proceed to the lymphodepletion on day -5. So that's a super important to orchestrate all the activities between the cell factory and the clinical team, so this whole process can go smoothly. The drug product is released, and all the documentation is also in place for that release. So in summary, when we look at different CMC challenges in initiating the clinical pivotal trials, they should not be underappreciated. So one needs to have a consistent and robust cell manufacturing process. This starts with the starting materials, goes through the analytical methods and the comparability. One needs to also assure that all the risks are minimized for the manufacturing failures. The out-of-specification products is a big challenge, so you need to determine, first minimize the risk for it, and then determine how you will act when you have such a product. Then the rigorous shipping procedures needs to be developed so that the product can be delivered reproducibly to the patient. And last but not least, I will repeat it for the end time comparability. So in conclusions, there are a few high-level learnings for the pivotal trial scale-up. First, robust CMC process, including comparability, is the key. Well-orchestrated release of the IMP to deliver fresh product with a short shelf life. Synchronize activities between CMC and clinical required to reproducibly deliver fresh product to eligible patients in compliance with regulatory requirements. And last but not least, advanced planning, close communication with the FDA is important to deliver adequate study protocol and also align with FDA on the CMC. Thank you.